Hello there, and welcome back to the Sleepy Nova Chronicles. 100 million years ago, a flower opened for the first time. Not the first flower in the absolute sense. Flowering plants had been evolving for tens of millions of years before this moment, developing their reproductive structures in incremental steps that the fossil record preserves only partially, in scattered impressions in ancient rock that tell the story in fragments rather than in sequence. But this particular flower, or one very much like it, in the warm, humid forests of what is now Southeast Asia, opened its petals around a structure that would change the trajectory of life on Earth more completely than almost any other single evolutionary development in the history of complex organisms. It offered something, not passively, not by accident, with a specific, targeted, evolutionary precision of a system that had been building toward this moment across millions of years of coevolution between plants and the insects that visited them. The flower offered nectar, a concentrated solution of sugars produced at significant metabolic cost by the plant, placed precisely where a visiting insect would have to brush against the plant's pollen to reach it, and it offered this nectar in the knowledge, encoded not in any conscious awareness but in the logic of natural selection, that the insect that took the nectar would carry the pollen to the next flower, and the cross-pollination that resulted would produce the genetic diversity. That gave the plant's offspring the best chance of surviving whatever the world produced next. The flower invented a contract, and one insect, more than any other, signed it. The bee. Not the bee you imagine when you picture the word. Not the single bee hovering at a garden flower on a summer afternoon, moving from petal to petal with the unhurried industry that most people find, instinctively and without quite knowing why, calming to watch. Not even the honeybee in its familiar striped form, which is itself a relatively recent development in a lineage that stretches back to that ancient forest. The bee that signed the contract 100 million years ago was something older and stranger, a wasp. A predatory wasp, from the same family that still produces the yellow jackets and hornets of the modern world, that at some point in the Cretaceous period made a transition so consequential that it effectively divided the history of flowering plants into before and after. It stopped hunting other insects and started eating pollen. This is the misconception to release tonight. The idea that bees have always been bees, that the gentle, flower-visiting, honey-producing organism you picture when you close your eyes was built from the beginning for the role it occupies, that the relationship between bee and flower is as old and as fixed as the landscape it takes place in. It is not. The bee is a wasp that retooled itself, that redirected the hunting apparatus of a predator toward the harvesting apparatus of a pollen collector, that converted the leg structures used to grip struggling insect prey into the leg structures used to carry pollen baskets, that converted the aggressive territorial chemistry of a wasp colony into the complex, regulated, cooperative chemistry of a bee colony that will, in a single summer, visit up to 50 million flowers to produce the few kilograms of honey it needs to survive the winter. 50 million flowers. One colony. One summer. That number sits inside every jar of honey you have ever opened. The bee's story is not a gentle story in the sense of being simple or small. It is gentle in the sense of being patient, the patience of an evolutionary process that took a predator and rebuilt it, over millions of years, into something that produces, as the primary output of its remarkable biological complexity, sweetness. That fills the landscape with a low, warm, continuous sound that most people, most cultures, across most of human history, have associated with abundance, with summer, with the specific sensory confirmation that the world is producing what it is supposed to produce, and that nothing, right now, is wrong. The hum of bees is the sound of the world working. You have known this your whole life, below the level of language. Your nervous system registered it before you could speak. The specific frequency of bee flight, between 200–250 Hz, falling precisely in the range of sound that the human auditory system processes as non-threatening, as ambient, as the acoustic signature of a productive landscape that poses no danger to the organism listening to it. Your nervous system hears bees and relaxes, not because of any decision you make, because the sound itself, that warm, low, continuous vibration, carries in its frequency a signal that your deepest biology interprets as safety. As the presence of flowers, which means the presence of fruit, which means the presence of food, which means the world is providing what is needed, and the season is good, and nothing urgent is required of you right now except to be present in a landscape that is doing its work. Detectives looking at the neurological response to natural soundscapes revealed that the sound of insects, including the flight frequency of bees, produces measurable reductions in heart rate and skin conductance in human subjects. Response is consistent with parasympathetic activation, the physiological state of calm and rest and digestion that is the opposite of the fight-or-flight response. The same neurological machinery that the forest's phytoncides activate through your respiratory system, the slow water activates through your auditory system, the sound of bees activates through the same pathway. The signal arrives below conscious awareness and produces, automatically, the physiological shift toward calm. You were built to find bees calming. Because the world your ancestors lived in was the same world the bees built, the flowering, fruiting, pollen-rich, honey-scented landscape that the bees' 100 million-year contract with the flower produced. The bees built the food supply, built the landscape, built, through their ceaseless pollination of the flowering plants that dominate the world's terrestrial ecosystems, the biological abundance that made complex animal life, including your life, possible in the forms it takes today. 1/3 of every mouthful of food you eat exists because a bee visited a flower. Not approximately 1/3. 1/3, the specific fraction of global food production that depends on insect pollination, the fraction that would not exist if the bees stopped flying, that would require the collapse of the agricultural systems that feed most of the human population to replace with any alternative that current human technology could provide. You are fed by bees, have been fed by bees, through the food chain, for as long as your lineage has eaten fruit or seeds or any of the other products of flowering plants that the bee's ancient contract with the flower made possible. And tonight, lying still in the dark, with the day finally beginning to release its hold on you, the biology that the bee built, the specific nutritional legacy of 100 million years of pollination, incorporated into the food that became your body, is doing its quiet, maintenance-level work in the dark of your cells. Tonight we are going to follow the bee. From the ancient wasp in the Cretaceous forest that first tasted pollen instead of prey, through the mechanics of how a bee navigates, how a hive thinks, how honey is made, how the colony survives the winter in a cluster of warmth in the dark, to the place where all of it arrives in you. In the sugars that fuel your brain. In the warmth that the hive produces and that your own body produces and that, right now, is settling into the specific, still, comfortable temperature of a body that has decided, finally, to rest. If this gentle journey is useful to you tonight, a slow hour of bees and deep time and the warm, humming, honey-scented patience of one of the most ancient and most consequential partnerships in the history of life, then I would gently invite you to subscribe, or to leave a thought below about which part of this stays with you. It helps other people who are lying in their own warmth and dark, also trying to let the day's buzz settle into quiet, find their way here. One flower at a time. But for now, there is only this. Allow your shoulders to soften. Let your breathing slow, not push toward any particular rhythm, just allowed to find the pace it finds when nothing is demanding speed. The pace of a bee moving between flowers on a warm afternoon. Unhurried, purposeful, going where the scent leads. Let your attention settle into the warmth of the hive, dark and close and full of the low, continuous vibration of 50,000 lives doing their patient, cooperative work in the sweet-smelling dark. The hive has been warm for 100 million years. Tonight it is warming you. Let's begin. A honeybee colony is not a collection of insects. It is a single organism. Not in the metaphorical sense that a team or a city or a corporation is sometimes called an organism. Not as a loose analogy for coordinated group behavior. In the strict biological sense of a system whose individual components cannot survive independently, whose collective behavior cannot be predicted from the behavior of any individual component, and whose physiological processes, temperature regulation, immune response, reproduction, sensory integration, decision-making, operate at the level of the whole, rather than at the level of any of its parts. The bee is a cell. The hive is the organism. This reframing is not a poetic embellishment. It is the framework that biologists who study social insects have converged on after decades of research into how colonies function. Research that has consistently revealed that the individual bee, taken out of its colony context, is as incomplete and as incapable of sustained function as a single neuron removed from a brain. The neuron has all the biological machinery of a cell. It can survive for a time in the right conditions. But it cannot think, cannot process information, cannot produce the outputs that make it useful, because the outputs are properties of the network, not of the node. The bee outside the hive is a neuron outside the brain, still alive, still functional at the cellular level, but missing the context in which its specific capabilities become part of something larger. The hive, by contrast, is capable of things that no individual bee could achieve or even approximate. It can regulate its internal temperature to within 0.5 degree Celsius of 35 degrees, regardless of the external temperature, maintaining the precise thermal environment that developing larvae require whether the world outside the hive is at -10 or +40. It can make collective decisions about where to move the swarm, which food source to prioritize, when to replace an aging queen, through a process of distributed information processing that involves thousands of individual bees exchanging information in ways that produce outcomes no single bee could calculate. It can remember. The hive maintains a spatial memory of the surrounding landscape, a distributed representation of the locations of food sources, water sources, and landmarks across an area of approximately 80 square kilometers around the hive entrance. That memory is not stored in any single bee's brain. It is stored in the population of foragers, each one carrying, in its individual memory, information about a subset of the landscape, contributing that information to the collective through the dance communication system when it returns to the hive, updating the collective's representation of the landscape as conditions change. 80 square kilometers. That is an area roughly equivalent to the city of Paris within its peripherique. Mapped, remembered, continuously updated by a colony of insects whose individual brains contain approximately 1 million neurons each, compared to the approximately 86 billion neurons in your brain. 1 million neurons per bee, 86 billion per human. And yet the colony, through the coordination of its 50,000 1 million neuron members, maintains a spatial memory and a decision-making capacity that, in terms of the sophistication of its outputs, has no obvious inferior status to the memory systems of much larger-brained animals operating on the same spatial scale. The monster of bee biology is not the individual bee, small, short-lived, operating on a brain the size of a sesame seed. The monster is the collective intelligence that emerges when 50,000 such brains are connected through chemical and behavioral communication into a system whose computational power is not the sum of its parts, but something that exceeds that sum in ways that the science of complex systems is still working to fully characterize. The waggle dance is the most remarkable communication system ever documented in a non-human animal. When a forager bee returns to the hive having discovered a rich food source, it does not simply go back to foraging. It performs. On the vertical surface of the honeycomb, in the dark interior of the hive, surrounded by hundreds of other bees whose antennae press against the dancer's body to feel the vibrations of the dance, the returning forager performs a figure-8 movement called the waggle dance, a precisely encoded communication that transmits to the watching bees the direction, the distance, and the quality of the food source it has found. The direction is encoded in the angle of the straight run at the center of the figure-8. The bee performs its straight run at an angle relative to vertical that corresponds precisely to the angle between the food source and the sun, as measured from the hive's location. If the food is directly toward the sun, the straight run points straight up. If the food is 90 degrees to the left of the sun, the straight run points 90 degrees to the left of vertical. The dance is a representation of solar geometry, performed in the dark, on a vertical surface, translated from the three-dimensional landscape outside the hive into the two-dimensional surface of the comb. The bee has invented a symbolic map. The distance is encoded in the duration of the waggle run. Approximately 1 second of waggling corresponds to approximately 1 kilometer of flight distance to the food source, with a relationship precise enough that experienced dancer watchers can use it to predict, within 100 meters or so, where the indicated food source is located. The quality is encoded in the vigor and duration of the entire dance. A particularly rich food source produces a longer, more energetic dance that recruits more followers than a mediocre source produces. Detectives looking at the waggle dance using high-speed cameras and electronic hive monitoring systems that tracked the subsequent flight paths of bees that had attended a dance revealed something that confirmed what the dance's information content suggested, but that the researchers still described as striking when they saw it directly. The attending bees flew to the location indicated by the dance. Not approximately. Not in the general direction. To the specific location, within the precision limits of the dance's encoding, indicated by the angle and duration of the waggle run they had attended in the dark interior of the hive. The dance works as a communication system, as an encoding of spatial information, as a mechanism for the collective allocation of foraging effort across the landscape, directing more bees to richer sources, withdrawing effort from depleted ones, updating the collective foraging strategy in real time as the landscape changes through the day and the season. It works with a precision and a sophistication that, when Carl von Frisch first deciphered it in the 1940s and described it to his colleagues, was met with skepticism so intense that it took decades of subsequent research, and eventually a Nobel Prize in 1973, to establish beyond reasonable doubt that a small insect was performing, in the dark interior of a wooden box, a symbolic communication that encoded spatial information in angular and temporal variables. A small insect. Symbolic communication. In the dark. Your own spatial memory. Your ability to navigate familiar environments, to know without consciously calculating that the coffee shop is 3 blocks north and 1 block east of the train station, to remember the layout of a building you visited once 6 months ago, is produced by a structure in your brain called the hippocampus. The hippocampus contains specialized neurons called place cells that fire when you are in a specific location, building a cognitive map of the environments you inhabit. That cognitive map can be updated, new information about changed layouts or new routes integrated into the existing representation, and it can be navigated during planning, allowing you to mentally simulate routes through environments before physically traversing them. The forager bee arriving at the hive entrance and beginning its waggle dance is doing something structurally similar, translating a cognitive map of the landscape into a communication that allows other bees to update their own maps without having visited the location. The bee and your hippocampus are solving the same problem, with different hardware, different scale, different medium of communication, but the same fundamental cognitive task: encoding spatial information, storing it, and sharing it with others in a form they can use to navigate the same space. Now consider what the hive does with the information those dancers produce, what the collective foraging effort, directed by thousands of individual dancers across the day, actually achieves in terms of the material it brings back to the hive. A single honeybee forager carries, on its return trip to the hive, a nectar load of approximately 40 milligrams, roughly the weight of a single small raindrop. 40 milligrams. Collected from between 50 and 100 flower visits in a single foraging trip, each visit taking between 1 and 2 minutes, the bee working continuously from the first light warm enough to permit flight until the failing light of evening signals the end of the foraging day. 40 milligrams per trip. And that nectar must be converted to honey before it provides the colony with stable, long-term energy storage. The conversion is not simple. Nectar is approximately 80% water and 20% sugar, a dilute solution that would ferment and spoil if stored in its raw form. Honey is approximately 17 to 20% water and 80% sugar, a concentrated, low water activity solution in which microbial growth is suppressed and indefinite storage becomes possible. To go from nectar to honey, the colony must evaporate approximately 80% of the water in the nectar it collects. It does this by passing the nectar repeatedly between bees, each bee holding the nectar in its honey stomach, exposing it to air through repeated regurgitation and re-ingestion in a process that spreads a thin film of nectar across the bee's mouthparts and allows water to evaporate, and by fanning the honeycomb cells with their wings to move air through the hive and carry away the evaporating moisture. The fanning. The sound of the hive that carries through the wood of the hive box on a warm afternoon, that deep, warm, continuous vibration that, detectives looking at the acoustic properties of beehives documented, falls primarily between 200–500 Hz and can be felt as much as heard when you place your hand against the hive's surface, is not simply the sound of flying. It is the sound of the colony processing its harvest, of thousands of pairs of wings moving in coordinated bursts to maintain the airflow that converts dilute nectar into the stable, sweet, aromatic concentrate that makes the colony's winter survival possible. The hum of the hive is the sound of chemistry in progress. To produce 1 kilogram of honey, approximately the volume of a standard honey jar, a colony must visit approximately 4 million flowers, fly a collective distance of approximately 40,000 kilometers, and process the collected nectar through the repeated evaporation and enzymatic conversion that transforms it from a dilute, perishable solution into the amber, viscous, virtually imperishable substance that fills the comb. 40,000 kilometers. The circumference of the Earth is approximately 40,000 kilometers. 1 jar of honey represents a collective flight distance equal to once around the planet. Your own body produces its primary energy currency, ATP, through a process of chemical conversion that is, in its fundamental logic, not entirely different from the hive's honey production. Your cells take in glucose, the simple sugar that your digestion extracts from the food you eat, and convert it through a series of enzymatic reactions into ATP, stripping energy from the glucose molecule in controlled steps and storing it in the phosphate bond of ATP, the universal energy currency of life that powers every biological process in every cell of your body. The hive converts dilute nectar into concentrated honey. Your cells convert glucose into concentrated energy. Both processes use enzymes. Both operate through sequential chemical transformations. Both produce, from a relatively dilute starting material, a concentrated, stable, long-term energy store that the system can draw on when direct energy input from the environment is not available. The hive stores energy for winter. Your cells store energy for sleep. Both are running their chemistry right now. Detectives looking at the temperature regulation of honeybee colonies revealed a biological thermostat of extraordinary precision. The brood nest, the region of the hive where the eggs and larvae develop, must be maintained at 34 to 35 degrees Celsius for the larvae to develop normally. Temperatures 1 or 2 degrees above or below this range produce developmental abnormalities. The colony maintains this temperature through the collective behavior of thousands of bees, clustering tightly over the brood to conserve heat when the temperature drops, fanning vigorously to cool the brood when it rises, moving water into the hive and fanning it to produce evaporative cooling in extreme heat. The regulatory precision is 0.5 degree Celsius, maintained across a temperature range of external conditions spanning from -20 to +40. Your own body maintains your core temperature at 37 degrees Celsius with a similar precision. Approximately 0.5 degree above or below this set point triggers physiological responses that correct the deviation. You sweat when you overheat. You shiver when you cool. Your blood vessels dilate toward the skin surface to shed heat or constrict toward the core to conserve it. Both systems, the hive and your body, are running the same thermostat, at different scales, with different mechanisms, producing the same output. A warm, dark, precisely regulated interior in which the most essential biological work can proceed. The hive is warm tonight. In the dark of its wooden box, in whatever corner of the world the nearest colony occupies, 50,000 bees are clustered in the winter ball, a sphere of warm bodies pressing together, the outer bees insulating the inner ones, the whole cluster generating heat through the slow contraction of flight muscles that produces warmth without movement, the metabolic furnace of 50,000 small bodies maintaining the interior temperature of the cluster at exactly the level the colony requires. to survive until spring. The hive is warm in the dark, as you are warm in the dark, both of you maintaining your precise interior temperature against the cold outside, both running your chemistry in the quiet, both exactly where you need to be. Of the 20,000 species of bee on Earth, only 7 make honey. 7. 20,000 species, found on every continent except Antarctica, in every habitat from Arctic tundra to tropical rainforest to suburban garden to the cracks in urban pavements, where the most adaptable solitary species have learned to nest in the thin soil between the concrete and the curb, and the one that most people picture when they hear the word bee, the one that lives in a wooden box and produces honey and dances in the dark to communicate the location of flowers, represents 7 species out of 20,000. The other 19,993 species live alone, not in colonies, not in hives with queens and workers and the elaborate social architecture of the honeybee superorganism. Alone, in tunnels in the ground, in hollow plant stems, in holes in wood, in small chambers excavated in clay banks, in the centers of flower heads, in whatever small, enclosed, protected space the individual female bee has found or made for herself and in which she will lay her eggs, provision them with pollen and nectar, seal the chamber, and leave, never to return, never to meet the offspring she has provisioned, never to experience anything that would be recognizable as the social life of the colony. Solitary, independent, silent, and collectively the most important pollinators on Earth. Not the honeybee. The solitary bees, in their 20,000 species of individual shapes and sizes and life strategies and flower preferences, are responsible for a greater proportion of the world's wild plant pollination than any managed honeybee population could provide. Because the honeybee, for all the extraordinary sophistication of its colony intelligence and its waggle dance navigation and its precise temperature regulation, is a generalist. It visits whatever flowers are producing nectar in the quantity and quality that the colony's needs demand. It is efficient. It is scalable. It is the agricultural standard because it can be moved, managed, and directed toward whatever crop needs pollinating at whatever time the farmer requires. But it is not specialized, and specialization is what the plant world needs most. Detectives looking at the relationship between solitary bee species and the plants they pollinate revealed a network of connections so specific and so intricate that it makes the waggle dance look like a rough approximation. Many plant species have evolved flowers so precisely matched to a single bee species. In their shape, their scent, the timing of their opening, the position of their pollen-bearing structures relative to the body of the bee that visits them, that no other pollinator can perform the service efficiently. The squash bee, Peponopis pruinosa, visits only plants in the genus Cucurbita. Squash, pumpkins, courgettes. Its entire adult life is organized around these plants. It times its emergence from its underground nest to coincide precisely with the opening of squash flowers, which open before dawn and close by mid-morning. The squash bee is airborne before sunrise, moving through the garden in the dark, finding flowers by scent alone before the light is sufficient to see them, collecting pollen with the specific body dimensions and the specific leg structures that have co-evolved with the squash flower's architecture over millions of years of mutual refinement. The squash bee works in the dark, before the honeybee has left the hive. And when the squash flower closes at mid-morning and the day's pollination work is done, the male squash bee does something that, when detectives first documented it using infrared cameras in squash fields at dusk, produced images so unexpected that the researchers checked their equipment. The male squash bee crawls into the closing squash flower and goes to sleep inside it. Not briefly, not as a resting pause between foraging flights. For the night. The male squash bee selects a closing flower in the early afternoon, crawls deep into the corolla as the petals begin to fold around him, and spends the night enclosed in the flower's warm, scented interior, emerging only when the flower opens again the following morning to resume its pollination role. The bee sleeps in the flower, and the flower that closes around the sleeping bee is not merely a convenient shelter. It is, in the specific chemical environment it creates inside the closed corolla, the warmth retained by the thick petals, the humidity maintained by the flower's transpiration, the specific blend of aromatic compounds that the flower produces in its interior, a space so precisely suited to the bee's overnight requirements that the relationship between the squash bee's sleeping behavior and the squash flower's closing architecture appears to be genuinely co-evolved. The flower produces a sleeping pod. The bee that pollinates it provides the next morning's service in exchange for a night's accommodation. The flower built a bedroom for the bee that keeps it alive. 100 million years of contract, renewed every night. Your own sleep environment, the specific combination of temperature and darkness, and the absence of disturbance that your nervous system requires to cross the threshold from wakefulness to sleep, was not always a mattress in a room with a door. For most of human evolutionary history, it was whatever the landscape provided. A hollow in a tree, the shelter of a rock overhang, the warmth of a group of bodies pressed together in the dark. The specific microclimate that the available environment created, not always comfortable, not always warm, but sufficient. The minimum conditions under which the biology of sleep could proceed. The squash bee has found its minimum conditions. It has found them with a precision refined over millions of years of evolutionary pressure, in a flower that has been building toward this specific accommodation for the same duration, in a relationship so intimate and so mutually beneficial that separating one from the other would, eventually, end both. The bee needs the flower to sleep. The flower needs the bee to reproduce. Everything holds. Now consider the bumblebee, the round, warm, slow-moving, generously furred member of the bee family whose flight has been described, in a famous and entirely incorrect popular claim, as aerodynamically impossible. It is not aerodynamically impossible. It is aerodynamically complex. The bumblebee's flight involves a wing stroke pattern and a body geometry that early aerodynamic models, designed for fixed-wing aircraft, could not accurately describe. When the correct model was applied, one that accounted for the low speed, high angle of attack, vortex-generating flight regime that the bumblebee actually uses, the aerodynamics worked perfectly. They had always worked perfectly. The bumblebee had been proving them correct for 30 million years before anyone tried to calculate them. The bumblebee is the most cold-tolerant bee on Earth. It is found at altitudes and latitudes where no other bee species can survive, in the Arctic tundra of Siberia and Alaska, at elevations above 4,000 meters in the Himalayas, in the high mountain meadows where the season for flowering is measured in weeks rather than months and the temperature can drop below zero on any night of the year. It survives these conditions through a physiological mechanism that is, in its operation, closely analogous to your own muscle warming response on a cold morning. Shivering. Before a bumblebee can fly in cold temperatures, it must warm its flight muscles to a minimum operating temperature of approximately 30 degrees Celsius, well above the ambient temperature in the Arctic habitats where it forages. It does this by decoupling its flight muscles from its wings and contracting them rapidly without producing movement, generating heat through the inefficiency of muscle contraction without the work of flight. A bumblebee warming up for flight in cold conditions vibrates. You can feel it if one lands on your hand on a cool morning, the small, warm trembling of a body generating its own heat before it is ready to fly. The bumblebee carries its warmth with it, generates it internally, maintains it against the cold of the environment through the insulation of its dense fur, the pile of branched hairs covering its body that traps air and functions, in thermodynamic terms, as a biological down jacket, and through the continuous generation of metabolic heat by the flight muscles that keep it warm even when it is not flying. Your own body generates its warmth in the same way. Your resting metabolism, the basal metabolic rate, the minimum rate of cellular energy consumption that maintains your body temperature without any additional physical activity, produces approximately 80 watts of heat. 80 watts. The output of a standard incandescent light bulb, generated continuously by the chemical reactions in your cells, maintaining your core temperature at 37 degrees against whatever the environment outside your skin provides. You are a warm body in a cold world, as the bumblebee is, both generating your warmth internally, both maintaining your precise interior temperature through the insulation of your covering, your skin and clothing, the bumblebee's fur, and the continuous metabolic heat of your biochemistry. Both warm in the dark, in whatever corner of the world tonight finds you. Detectives looking at the navigational abilities of bumblebees in environments where the researchers had systematically altered landmarks revealed something that connects the bumblebee's cognitive abilities to the bee's wider story of patient, accumulated, evolutionary intelligence. Bumblebees form cognitive maps of their foraging territory, representations of the spatial layout of flower patches, landmarks, and routes that they update as the landscape changes, and that they use to plan efficient foraging routes rather than simply following a fixed sequence of waypoints. The bumblebee optimizers. When researchers presented bumblebees with a set of artificial flowers at known locations and tracked their foraging routes over multiple days, the bees converged on routes that were close to the mathematically optimal solution to what computer scientists call the traveling salesman problem. The problem of finding the shortest route that visits a set of locations exactly once. The traveling salesman problem is computationally difficult for the same reason that the bumblebee's foraging route is impressive. The number of possible routes grows exponentially with the number of locations to visit, making an exhaustive search for the optimal solution impractical, even for powerful computers, as the number of locations increases. The bumblebee solves it approximately, efficiently, in real time, on a brain the size of a grass seed. Not because it is performing mathematical calculation. Because the evolutionary pressure of minimizing energy expenditure while maximizing nectar collection has shaped, over millions of years of refinement, a foraging strategy that happens to approximate the optimal solution. The way the evolution of the bird's wing happened to approximate the optimal aerodynamic form for low speed, high maneuverability flight without any engineer ever calculating what that form should be. Evolution finds good solutions, given enough time, and the bee has had enough time to find the squash flower's bedroom, enough time to generate the warmth that the Arctic meadow demands, enough time to solve the traveling salesman problem in the space of a few foraging days, enough time to build, across 20,000 species and 100 million years of coevolution with the flowering plants that now dominate the terrestrial world, a pollination network so dense and so redundant and so precisely matched to the needs of the plants it services that the landscape itself, every forest and every meadow and every hedgerow and every patch of urban wildflower, is, in a measurable ecological sense, the bee's creation. The bee did not evolve to fit the landscape. The bee built the landscape. And tonight, in the dark outside your window, wherever the nearest patch of unmown grass or hedgerow or garden border lies within range of a sleeping bee's territory, the evidence of that building is present. In the seed heads of the plants that will open next spring. In the soil below the patch of bare earth where the mining bee's tunnel entrance marks the location of a nest provisioned with pollen and sealed with clay, containing the eggs that will hatch next year and emerge as the next generation of pollinators who will spend their brief adult lives doing exactly what their parents did, in exactly the same patch of ground, visiting exactly the same plants, maintaining the same intimate, ancient, patient contract between flower and bee that began 100 million years ago in a forest that no longer exists. The contract continues, in the dark, in the warm, in the close, sweet, still air of the sleeping hive and the sleeping flower and the sleeping bee curled in the corolla, waiting for morning. As you are waiting, not urgently, not with any awareness of what comes next, just allowing the dark to hold you in the same way that the closing flower holds the sleeping bee, completely, without reservation, for exactly as long as is needed. In the winter of 2006, beekeepers across the United States began opening their hives to find them empty. Not dead. Empty. The bees were gone, not lying on the floor of the hive box in the way that a colony dying of cold or starvation dies, with the accumulated bodies of the winter cluster providing at least the evidence of what had been there. Simply absent. The hive boxes contained honey, contained capped brood, contained the physical infrastructure of a functioning colony, and contained almost no adult bees. The foragers had flown out and not returned. The hive had been evacuated, not in the way that a swarm evacuates, with a deliberate, organized, pheromone-coordinated departure of a reproductive swarm carrying a queen, but in a way that left no trail, no cluster, no indication of where the bees had gone or why. The phenomenon was named colony collapse disorder, and it was not new. Historical records of similar mass disappearances of honeybee colonies stretch back centuries, to accounts in European beekeeping literature of the 18th and 19th centuries describing hives that emptied without explanation, to even earlier records that may describe the same phenomenon under different names in different languages. But the scale of the 2006 event, and the events that followed it across subsequent winters, was unprecedented in modern beekeeping history. In the United States alone, between 2006 and 2011, approximately 30% of managed honeybee colonies were lost each winter, a loss rate that, in some regions, reached 50% in a single winter. 30%. Every winter. For 5 consecutive years. A managed honeybee colony represents approximately 2 years of work to establish from a new package of bees to a productive, wintering colony. Losing 30% of those colonies every winter means replacing them at a rate that, for many commercial beekeepers operating on the margins of profitability, was not sustainable. The commercial beekeeping industry in the United States contracted. The number of managed colonies fell, and the agricultural systems that depended on those managed colonies for pollination, the almond orchards of California, the blueberry fields of Maine, the apple orchards of Washington State, found themselves competing for a shrinking supply of the pollination services that their crop yields depended on. The cause of colony collapse disorder is not a single thing. It is a convergence, a catastrophe assembled from multiple simultaneous pressures, each one insufficient alone to produce the collapse, but together creating a burden on the honeybee colony that exceeds what the colony's biology evolved to carry. The Varroa mite. Varroa destructor. The name is not accidental, is a parasitic mite that attaches to the bodies of honeybee larvae and adult bees, feeding on their fat bodies, the biological equivalent of the liver, the organ that processes nutrients and produces the proteins that sustain the bee through winter, and transmitting viral diseases as it feeds. The mite arrived in Western honeybee populations from Asian honeybee populations in the late 20th century, carrying with it a suite of viruses to which the Western honeybee had no evolved immunity. It spread globally within decades. A single Varroa mite on a developing larva can reduce the adult bee's lifespan by 20 to 30%, can reduce the protein content of its fat body by 50%, can impair its navigation, its learning, its immune function, can introduce viral infections that further compromise every system the bee depends on to perform its role in the colony. Detectives looking at the relationship between Varroa infestation levels and colony survival through winter revealed a threshold effect. Colonies with Varroa mite levels below approximately 2 per 100 adult bees in late summer can survive the winter with normal management. Above that threshold, the probability of winter collapse increases steeply, the relationship between mite load and colony failure following a curve that maps, in biological terms, the point at which the cumulative damage to the colony's winter bees exceeds the colony's capacity to compensate. The mite finds the threshold, then crosses it. Your own immune system operates on a similar threshold logic. The pathogens that circulate through your environment are present at all times. In the air you breathe, on the surfaces you touch, in the food you eat. Your immune system contains them, prevents them from reaching the concentrations that produce illness, and does so without your awareness as long as the system is functioning at adequate capacity. When the system is compromised, by inadequate sleep, by chronic stress, by nutritional deficiency, the threshold shifts. The same pathogen load that your well-rested immune system manages without producing symptoms becomes the pathogen load that your depleted immune system cannot contain. The bee and your immune system are both threshold systems, both managing a load that is always present, both vulnerable when the capacity to manage it drops below the level the load requires. But the varroa mite is only one of the pressures the bee is carrying. The landscape it forages in has been changing. The agricultural intensification of the 20th century converting the flower-rich, mixed farming landscape of hedgerows and wildflower meadows and varied crop rotations into monocultures that produce large quantities of a single crop and almost nothing else. A bee foraging in a monoculture landscape, a landscape of wall-to-wall oilseed rape or sunflowers or almonds, has access to enormous quantities of one type of pollen for a brief period while the crop flowers, and then almost nothing for the rest of the season. This is a nutritional problem. Bees, like humans, require a diverse diet. The pollen of a single plant species provides only a fraction of the amino acid profile that a developing bee larva needs. A larva provisioned exclusively with monoculture pollen develops with nutritional deficiencies that impair its immune function, its learning capacity, and its longevity. A colony foraging in a diverse, wildflower-rich landscape produces workers that live longer, navigate more effectively, and resist disease more successfully than a colony foraging in a monoculture, because the diverse pollen provides the complete nutritional profile that the bee's biology evolved to receive from a landscape that offered dozens of plant species in flower simultaneously. The bee evolved to forage in diversity, and the modern agricultural landscape has removed the diversity. Detectives looking at the pollen loads carried by forager bees in landscapes of different agricultural intensity revealed that bees in intensively farmed landscapes carry less diverse pollen than bees in mixed or organic farming landscapes, a result so consistent across multiple studies in multiple countries that it has become a standard metric for assessing the quality of agricultural landscapes for pollinators. And then there are the neonicotinoids, a class of systemic insecticide. Insecticides that are absorbed by the plant from the soil and distributed throughout its tissues, including its pollen and nectar, that became the most widely used agricultural insecticides in the world in the 1990s and 2000s, applied to the seeds of major crops before planting so that every part of the developing plant would carry a dose of insecticide sufficient to kill the insects that fed on it, including, at sublethal doses, the bees that fed on its pollen and nectar. Sublethal doses do not kill bees outright. They impair them. Detectives looking at the effects of neonicotinoid exposure on honeybee behavior revealed impairments to navigation. Bees exposed to field-realistic concentrations of neonicotinoids were less able to return to the hive after foraging flights, showed reduced ability to learn floral scents, and performed waggle dances with reduced precision. The dance that encodes the spatial information that directs the colony's foraging effort. The dance whose precision Carl von Frisch spent decades documenting and that won the Nobel Prize, became less precise when the dancer had been exposed to neonicotinoids at concentrations present in the pollen of treated crops. The dance became corrupted. The information became less reliable. The collective intelligence of the hive depended on the accuracy of the individual dance. And the individual dance was being degraded by chemistry that the bee had no evolutionary preparation for and no biological mechanism to detect before it had already been absorbed. The bee had been solving the traveling salesman problem on a grass seed-sized brain for 30 million years, and it could not detect the specific class of neurotoxin that modern agriculture had placed in its food. You cannot detect it either. The same class of compounds, or their metabolic products, has been detected in the urine of a significant percentage of the general population in countries where neonicotinoids are widely used. You absorb them through the food chain, at concentrations far below those that produce acute toxicity in humans. The long-term effects of chronic low-level neonicotinoid exposure in human beings are not yet fully characterized. The research is ongoing, the data accumulating, the picture not yet complete. But the bee and you are both downstream of the same chemistry, both absorbing, through the food and the environment, compounds that arrived in the agricultural landscape within the last few decades, and that neither your biology nor the bee's biology has had time to develop any evolved response to. Both navigating a chemical landscape that is newer than either of your evolutionary histories. And here, In the convergence of the varroa mite and the nutritional poverty of the monoculture and the neurotoxic chemistry of the systemic insecticide is where the catastrophe reveals its specific character. Not a single overwhelming force. A slow accumulation of burdens, each one manageable alone, together exceeding the threshold. The same kind of catastrophe, it turns out, that the bee has survived before. The bee lineage has been through 5 mass extinctions, has survived the asteroid impact that killed the dinosaurs 66 million years ago, the impact that darkened the sky and cooled the planet and eliminated approximately 75% of all species on Earth, including most of the flowering plants that the bee depended on, has survived the ice ages that covered much of the northern hemisphere in glaciers and compressed the bee's habitat into the narrow refugia that remained ice-free at lower latitudes, has survived the previous periods of rapid climate change and vegetation shift that punctuate the geological record. It survived them through the same mechanism that has sustained it for 100 million years. Diversity. 20,000 species, distributed across every habitat the planet offers, each one specialized for different conditions, different plants, different climates, means that whatever catastrophe eliminates some species will be unlikely to eliminate all of them. The bee's diversification across 20,000 species is its insurance policy, its redundancy, its evolutionary hedge against the kind of single-point failure that would destroy a less diverse lineage. And that insurance policy is being called on right now. Because while the honeybee, the managed, commercial, agricultural standard, is under extraordinary pressure from the convergence of threats we have described, the solitary bees are responding differently. Many solitary bee species are resilient to the varroa mite, which only parasitizes the social bees. Many are less exposed to neonicotinoids because they forage on wildflowers rather than agricultural crops. Many are benefiting from the restoration and rewilding efforts that have been creating new habitat in the margins of agricultural landscapes. And the gardens are helping. Detectives looking at the diversity and abundance of wild bee species in urban and suburban gardens compared to the surrounding agricultural landscape revealed something that city dwellers and gardeners have been contributing to without necessarily knowing it. Urban and suburban gardens, diverse, flower-rich, pesticide-light, offering a continuous succession of blooming plants from early spring to late autumn, support higher densities of wild bee species than the monoculture agricultural landscapes that surround many cities. The garden outperforms the farm. For bees. Not in crop yield. Not in any metric the agricultural system measures, but in the ecological service that the bee provides and that the landscape requires, the pollination, the biodiversity support, the maintenance of the plant communities that sustain the food web. The garden, with its casual diversity and its absence of systemic pesticides and its willingness to let things bloom that agriculture would mow, provides something that the farm cannot. A window, a gap in the catastrophe through which the bee can pass. The bee has been finding gaps for 100 million years, finding them in the cracks of the geological record, in the refugia of the ice ages, in the margins of the mass extinction events that cleared the landscape and left the bee, patient, diverse, specialized, adapted, to fill it again. The bee knows how to find gaps. And right now, in every garden that has left a patch of clover unmown, in every urban verge that has been planted with wildflowers, in every allotment border where the weeds have been allowed to flower before they are pulled, the bee is finding the gap, flying through it, in the warm, in the hum, in the low, continuous 200 Hz vibration that your nervous system hears as safety, as abundance, as the world providing what it is supposed to provide. The bee is still there. It is still there. Your body knows. You can feel it in the calm that the thought produces, not the thought of the threats, but the thought of the bee finding its gap, as it has always found its gap, in the patient, unhurried, flower-by-flower way that a 100-million-year-old contract gets renewed. Petal by petal, season by season. Still, there is honey in your blood right now. Not literally honey, not the amber, viscous, aromatic concentrate that the hive produces from 40,000 kilometers of collective flight. But the sugars that honey is made of, the glucose and fructose that the bee extracts from nectar and concentrates into honey, and that you extract from the food you eat and concentrate into the blood glucose that fuels every cell in your body. The same simple sugars, derived, in their ultimate origin, from the same solar energy that powers the plant's photosynthesis and the bee's flight, and the entire biological chain that begins with a flower opening and ends with the sweetness in your blood tonight. The bee made this possible. Not alone. Not as the sole architect of the food system that feeds you, but as the essential link, the mobile, winged, pollen-carrying link in the chain between the flower's pollen and the fruit seed that makes the fruit possible, that makes the seed possible, that makes the plant's reproduction possible, that makes the next generation of flowering plants possible, that makes the nectar that the bee collects that becomes the honey that fuels the bee that pollinates the next flower. The chain has no beginning you can point to. It has been running for 100 million years without a break. And right now, tonight, the glucose in your blood, the fuel that your brain is running on as it processes these words and finds them, perhaps, a reason to stop processing anything else, arrived in you through that chain. Through the food that the bee's pollination made possible. Through the fruit or the seed or the vegetable that a bee visited at some point in its brief adult life, transferring pollen from one flower to another in the unhurried, purposeful, flower-by-flower way that the contract between bee and plant has always been executed. You are downstream of the contract. You have always been downstream of the contract. And here is the specific, biochemical, traceable way in which the bee's work lives inside you right now. Your brain, the 3-pound organ in your skull that is, at this moment, cycling through the early stages of the transition from wakefulness to sleep, consumes approximately 20% of your body's total energy budget despite representing only 2% of your body weight. That energy comes from glucose, from blood sugar, from the steady supply of simple sugars that your digestive system extracts from the food you eat and your liver regulates into the bloodstream at the concentration your brain requires. Your brain runs on flour sugar, processed by a bee, converted into fruit, eaten by you, extracted by your digestion, delivered to your brain by your blood, used right now to maintain the electrical activity of the neurons that are, at this moment, beginning their nightly transition from the fast, high-amplitude waves of waking consciousness to the slow, deep waves of restorative sleep. The bee's work ends in your neurons, in the specific, patient, sweetness-derived energy that is, right now, powering the last waking thoughts of a body that has decided, finally, completely, to rest. Detectives looking at the specific carbohydrates in honey revealed that honey contains not only glucose and fructose, but approximately 200 other compounds, enzymes, organic acids, antioxidants, aromatic molecules, that the bee adds to the nectar during the conversion process. The enzyme invertase, secreted by the bee's hypopharyngeal glands into the nectar during processing, splits the sucrose in nectar into glucose and fructose, making the sugars more immediately available to any organism that consumes them. The enzyme glucose oxidase produces gluconic acid and hydrogen peroxide as it acts on glucose, the hydrogen peroxide being responsible for honey's antimicrobial properties, the property that allows honey sealed in a comb cell to remain edible for thousands of years. Honey found in Egyptian tombs 4,000 years old was still edible. 4,000 years. Sealed in wax. Produced by the same biological process that produced the honey in your kitchen. Preserved by the same enzyme chemistry. Unchanged by 4,000 years of sitting in the dark of a sealed chamber, while the civilization that placed it there rose and fell, and was covered by sand, and excavated by archaeologists who found it, opened it, and tasted it, and found it sweet. The bee's chemistry outlasts civilizations. Your own body contains preservative chemistry that operates on the same principle as the bee's hydrogen peroxide system. Your immune system produces reactive oxygen species, hydrogen peroxide among them, as part of its antimicrobial arsenal. The same molecule, used for the same purpose, in a different system, at a different scale, in the body of a different organism. But the chemistry is the same because the problem is the same: preventing the proliferation of microorganisms in a biological medium that is rich in nutrients and therefore attractive to everything that eats nutrients. The bee solved this problem in honey. Your immune system solves it in your tissues. Both running variations of the same ancient oxidative chemistry that life first developed in the early ocean, in conditions that bear no resemblance to a bee's honey stomach or your bloodstream, but that established the biochemical toolkit from which both solutions were eventually assembled. You and the bee are running the same chemistry, in different forms, in different bodies, in the same dark, on the same night, with the same ancient molecular toolkit that life has been using since long before either of your lineages existed. Now consider what the bee does at night. The hive at night is not a hive at rest in the way that a sleeping mammal is at rest, metabolically reduced, sensory systems dimmed, consciousness suspended. The hive at night is a hive managing the transition from the day's foraging activity to the night's maintenance activity, maintaining its cluster warmth, processing the day's nectar harvest, producing wax for new comb construction, feeding the developing larvae that cannot wait for morning, monitoring the hive's internal chemistry through the pheromone signals that pass continuously between bees. The hive does not sleep, but the individual bees within it do. Detectives looking at the sleep behavior of individual honeybees using continuous video monitoring and measurement of antennal response to stimuli revealed that individual bees cycle through sleep states, periods of reduced movement, lowered antennae, reduced response to stimuli, that show measurable similarities to the sleep stages of vertebrate animals. Young bees sleep in shorter, more frequent bouts. Older forager bees sleep in longer, more consolidated periods, often in the dark interior of the hive away from the busier areas near the entrance. Some bees sleep in flower heads, folded into the petals, still and warm, in the same intimate shelter that the squash bee uses as its overnight accommodation. The bee sleeps like you, not with the same neurological complexity, not with the same sleep architecture, the specific cycling through REM and non-REM stages that characterizes vertebrate sleep and that produces, among other things, the consolidation of the day's memories into long-term storage. But with the same fundamental biological purpose: the reduction of activity, the restoration of depleted neurotransmitters, the repair of cellular damage accumulated during the day's work, the preparation of the nervous system for the next day's demands. A forager bee that has been sleep-deprived performs a less accurate waggle dance the following day. The dance that encodes the direction and distance of the food source, the dance that took 30 million years to evolve and that Carl von Frisch spent decades deciphering, becomes less precise when the dancer has not slept. The angular encoding drifts. The duration encoding becomes less consistent. The information that the colony depends on to allocate its foraging effort becomes less reliable. The bee's collective intelligence depends on the individual bee's sleep. As your collective function, your capacity to maintain the relationships and responsibilities and the social and professional roles that constitute your participation in the human community you are part of, depends on your individual sleep. The quality of your decision-making, your emotional regulation, your memory consolidation, your immune function, your physical coordination, all of these degrade in measurable, documented, reproducible ways when your sleep is insufficient. The individual bee's sleep deprivation degrades the collective intelligence of the hive. Your sleep deprivation degrades your capacity to be a functioning node in the human networks you inhabit. Both systems, the hive and your life, depend on the individual doing its maintenance work in the dark. Tonight is your maintenance work. Tonight is the dance being consolidated for tomorrow. Tonight is the 40,000 kilometers of accumulated flight being processed into the honey of tomorrow's function. The clear, concentrated, precisely encoded readiness for whatever tomorrow requires, produced from today's dilute, water-heavy harvest of experience and effort and information and emotion. The nectar of today becomes the honey of tomorrow. But only if tonight happens. Only if the bee returns to the hive. Only if the hive maintains its warmth in the dark. Only if the individual bee sleeps. If this journey has been useful to you tonight, if this slow hour of bees and flowers and the warm, humming, sweet-scented patience of 100 million years of contract between the living world and its pollinators has helped the day's nectar begin its conversion to something more stable and more sweet, then I would gently invite you to subscribe, or to leave a thought below about which part of this stayed with you longest. Whether it was the squash bee sleeping in the closing flower, or the winter cluster generating its warmth in the dark, or the bee finding its gap through the catastrophe with the same patient efficiency it has brought to every gap for 100 million years, or simply the knowledge that the glucose in your brain right now arrived there through a flower that a bee visited. It helps other people find their way here. Other people in their own warm dark, also processing the day's nectar into tomorrow's honey, also needing the hive's warmth to do it. One flower at a time. And if sleep is still arriving slowly, if the day's nectar is still too dilute to settle, there is another journey waiting. Another corner of the living world. Another deep time story told at this same unhurried pace, in this same quiet voice, carrying you somewhere warm and old and patient and larger than whatever is still buzzing at the edge of your attention. It will be there when you need it. But for now, feel the warmth of your body where it meets the surface beneath you. Not the warmth you produce against the cold, the warmth you simply are. The 80 watts of metabolic heat that your cells generate continuously, that your skin and your clothing and the air immediately around you collect and hold, that makes the space around your body in the dark a microclimate as specific and as deliberately maintained as the interior of the winter cluster. You are the cluster. You are 50,000 lives generating their warmth in the dark against the cold outside, except the 50,000 lives are your cells, and the cold outside is the temperature of the room, and the warmth they generate is the specific, precise, biochemically maintained warmth of a body that knows what it needs and has always known how to produce it. Allow your shoulders to soften now, the way the bee cluster softens in the warmth of its own making, not collapsing, not losing its structure, just releasing the tension that cold creates, settling into the shape that warmth allows when nothing is pressing against it. Allow your jaw to unclench. Allow your hands to open. Allow the thoughts that are still buzzing at the edge of your awareness, the last foragers of the day, still circling the hive entrance, still carrying whatever they gathered in the final hour of light, to land now, to come in, to join the cluster in the warm dark interior, where the night's work will convert them into something you can use tomorrow. Let them in. Let the entrance close. The hive is warm. The dance is done. The nectar is in the cells. 100 million years ago, a flower opened, and something came. Something patient and warm and precise, and connected to every flower that had ever opened and every flower that would ever open, through an unbroken chain of visits and pollen and sweetness that has been running without interruption across every catastrophe the earth has produced. It came. It did its work. It returned to the warmth in the dark, as you are returning now, to the warmth, to the dark, to the sweet, still, unhurried interior of a night that is asking nothing of you except this: rest. The contract is kept. The flower has been visited. The honey is forming in the dark. Sleep well, and good night.